Looking at patients with CLL, seeing that they suffer from an immune dysfunction, knowing that both the T cell compartment, the normal B cells, and also the neutrophil or myeloid compartment suffers from dysfunctional immune function. It's close to look next for what actually is the major cellular compartment in our body, the gut microbiome. We know that the gut microbiome has a direct impact on immune function. We know that changes in gut microbiome, at least in murine models, can impact the immune system, but also impact the development of malignancies far away from the gut. So we took a cohort of patients with CLL and additional cohorts of patients receiving hematological stem cell transplant for other hematological malignancies, patients going through thoracic surgery, and healthy controls, and tried to identify a signature in the microbiome that correlated with the event of having CLL. We have demonstrated first in the pilot study and now in the larger study that we can actually identify CLL patients as compared to healthy controls. So why go the next step, include other disease states? That's because we want to identify whether we found a signature just for a diseased microbiome. So it could be that all different types of diseases actually correlate with a dysfunctional microbiome. And to address that, to see if it's actually specific for CLL, we needed the other disease stages. And what we realized now by implementing machine learning methods to analyze these different microbiomes is that we can identify a higher likelihood of being CLL than other heme malignancies or patients going through surgery or healthy controls, but it's not 100% capability of actually identifying whether it's a CLL or one of the other disease states, whereas it's quite easy to identify whether it's CLL or healthy controls. This means that the microbiome of different diseases are actually closer to each other, but it also means that we can identify factors in the microbiome, different patterns of bacteria being represented, or lack of diversity, meaning that you have a more narrow microbiome with just a few species of bacteria dominating, that we could intervene on. So essentially the next steps would be both based on murine studies and hopefully eventually on human studies to see if we can change the microbiome. That could be by fecal transfer, it could be by changing the diet, or it could be by probiotics or antibiotics, to change the microbiome in a way where it could actually improve the immune function for patients with CLL and maybe eventually delay or prohibit the development of CLL into clinical disease needing treatment.